All right, morning everybody. I'm Alan Carr. Welcome to the 18th annual Needham Healthcare Conference. Very excited to have Cara Therapeutics with us today. We're going to have Derek Chalmers, CEO, present, give an update on the company and as well as an overview of pipeline, that sort of thing. And as Derek will discuss, they've got some data coming up soon. So, Derek?
Thank you, Alan. Good morning, everybody. This one on? Thanks for coming along this morning. I'm going to make some forward-looking statements during the next 20-25 minutes, of course. So today, Cara is a company focused on the development of a novel class of peripherally acting kappa agonist therapeutics, specifically for the treatment of systemic pruritus across multiple clinical populations. And we have two main development programs we're working on, as indicated here on slide number 3. So we have KORSUVA injection program, which is an intravenous formulation of our lead kappa agonist, CRA45, which I'll cover in just a second, which is in phase 3. We're currently running 2 phase 3 trials, one in the U.S., one global, for the treatment of chronic kidney disease-associated pruritus, specifically in hemodialysis patients. Here we have the CAM1 US trial, which is now fully enrolled, and we've guided we'll read out top-line data this quarter, Q2 of this year. We have a second pivotal phase 3 trial, CAM2, which is global, which we'll read out in the second half of this year. We have breakthrough designation for this compound, for this indication. We do intend to commercialize KORSUVA injection in the U.S., but we already have in place various regional partnerships, the most recent of which was Vifor Fresenius that we announced last year for the EU and select territories, and we have partners also in Japan and South Korea. So that's our first and lead program. Our second program is oral KORSUVA, an oral formulation of our lead kappa agonist. And here we're pursuing this formulation across multiple clinical populations where chronic pruritus is a significant unmet need. Currently, we have an ongoing phase 2b dose-ranging study in CKD-associated pruritus in pre-dialysis patients. This is a stage 3 to 5 CKD patients. And that is going to read out in the second half of 19. That's enrolling well, and I'll talk about that a little later on. Behind that, we have 2 other patient populations we're interested in for our oral program. Chronic liver disease-associated pruritus, we're about to initiate a phase 2 this quarter. And finally, atopic dermatitis, where we guided last month that we expect to initiate a phase 2 around about mid-year. So I want to take you through each of those programs in a little more detail. Before I do that, just on slide 4, a little reminder of the characteristics of our lead kappa agonist, CRA45, that underlie really its clinical profile. First and foremost, this is a first-in-class novel chemotype, as illustrated here on slide 4. Not a classical organic heterocycle, but an all-D-amino acid tetrapeptide. And that endows some specific advantages to the molecule, which you'll see as we run through the clinical data. 3 main advantages, the first being pharmacologically. We emulate the endogenous ligand for the kappa receptor, which of course is peptidic dynorphin. And you can see from the inset table here, we have exceptionally high cell activity specifically for the human kappa receptor. No interactions whatsoever with other human opioid receptors, the mu or the delta, and no interaction with any other GPCRs in the genome or transporter system. So we produce a very pure kappa agonism, really lacking in any other opioid side effects, including, importantly, euphoria and abusability, and I'll cover that a little later on in the presentation. The second and third major advantages really are PK and distribution and elimination. With this structure, this peptidic structure, it's fairly obvious we have a high degree of charge there, high polar surface area. This molecule inherently doesn't cross lipid bilayers, so we don't see entry across the blood-brain barrier. So we have a very peripherally restricted molecule and really a peripherally acting molecule at the kappa receptors. finally, this molecule is not metabolized via the liver, no CYP450 interactions whatsoever. It's excreted whole via the kidney. So there's minimum potential for drug-drug interaction with this molecule. So how does it work in pruritus, as illustrated in slide 5? Pruritus really emanates in the epidermis, or the dermal-epidermal junction, and it's relayed by a subset of sensory afferent C fibers, probably a subset of unmyelinated fibers, via the dorsal root ganglia and up into the brain. And these fibers are sensitive to locally released pruritogens that sensitize the nerves, mostly immune molecules that come from mast cells, macrophages, and other endogenous immune molecules. So KORSUVA, as illustrated in slide 5, acts on kappa receptors directly on these nerve endings, inhibits activity, so we dampen the activity directly of the neuronal fibers, but it also importantly acts on immune cells. So we can block the release of these immune cytokines that come from these particular cells endogenous to the epidermis and the dermis. So there's a dual mechanism here, and we believe that dual mechanism underlies the broad potential for this particular molecule to be antipruritic across clinical populations, really regardless of the initiating pathophysiology. And the opportunity across clinical populations is broad. So on slide 6 here, we're looking at a range of clinical conditions in which chronic systemic pruritus is a serious issue and an unmet clinical need. And these really range from perhaps the more obvious dermatological conditions such as atopic dermatitis, through systemic diseases such as chronic kidney disease and chronic liver disease, and all the way to neuropathic and psychogenic conditions. All of these indications can be characterized by severe pruritus. If we look at IMS numbers for the U.S., approximately 20 million scripts are written in the U.S. for the treatment of pruritus, simply for generic agents every year, predominantly corticosteroids. So there is a very large unmet need across clinical populations. Slide 7 is a snapshot of where we are in our development pipeline and how we're attacking each of these clinical populations with KORSUVA or KORSUVA injection. Obviously, our lead program being KORSUVA injection for phase 3, and then following that, all of our oral endeavors in phase 2 for CKD, chronic liver disease, and finally, atopic dermatitis. So I'm going to walk through each of those programs in a little more detail as to status and where we are and upcoming milestones. I'm going to start with KORSUVA injection for the treatment of CKD, AP, and dialysis patients. So what is this condition? Well, this is a very serious, consistent, systemic itch condition that occurs with high frequency in hemodialysis patients, up to 70%. Very serious condition that impairs quality of life across multiple aspects of quality of life, social, emotional, physical, is highly correlated with increased frequencies of infections, both local and indeed systemic, is correlated with increased hospitalizations for hemodialysis patients with severe pruritus, and is correlated with increased mortality risk for these patients expressing moderate to severe pruritus. So a severe unmet need, opportunity within this patient population, as outlined in slide 10, is large. There are approximately 500,000 hemodialysis patients in the U.S. 60 to 70% of these patients are expressing symptoms related to pruritus, and 40 to 50% of those patients are expressing moderate to severe symptoms. So no approved therapies. We aim for KORSUVA injection to be the first therapy with a label for this particular patients. From a commercialization standpoint, they are hemodialysis patients. Reimbursement does fall under the ESRD bundle. We've benefited in the last year from changes to that legislation, and we now know that this drug will be priced at ASP for the first 2 years after launch minimally to assess usage. And then we are in constant conversation with the CMS as to reimbursement options beyond that 2-year TDAPA period for innovative therapeutics, as we have with KORSUVA injection. So overall, where are we with the program? As I mentioned initially, we have 2 phase 3 readouts coming this year, the first this quarter, the second half of the year. We announced in January of this year that for CAM1, our initial U.S. phase 3 trial, we completed an interim conditional power assessment. This allowed us to use an IDMC to assess the maintained power to see a difference between our 845 treated patients and placebo, with 50% of those patients through the treatment period. So that was an unblinded analysis from our IDMC that would allow us to adjust sample size if we didn't see appropriate power to see a statistical difference. The advice we had from that, and we announced as public from that analysis, was not to increase the sample size, and consequently this trial was fully enrolled at the very beginning of January of this year. So again, we expect readout in this quarter. Commercialization, we have a partnership specifically on this formulation with Vifor Fresenius, and I'll cover the details for that particular partnership, which we think is an important one for the commercialization of this particular therapeutic product a little later. Another aspect in slide 12, I want to cover another advantage of KORSUVA injection related to chemistry and chemical structure. Being a tetrapeptide which isn't metabolized and excreted whole via the kidney, when we administer this compound intravenously post-dialysis, we really see a depot-like exposure for the compound for the next 2 days, essentially. So the patient benefits from the antipruritic action of the drug after 1 injection post-dialysis. Very convenient for the patient. These, of course, our patients are heavily pill-burdened with their comorbidities, mainly diabetes and hypertension. So that's a particular advantage. Also, before we get to the data, as illustrated in slide 12, even at the lowest dose we've used in our phase 2 trial, which is 0.5 micrograms per kilo, the blue line here, you see we have an exposure which is well in excess of that required for full human kappa receptor activity, illustrated by the dotted orange line on slide number 12. So all of the dosages we've used in our phase 2b trial are essentially supramaximal in terms of kappa receptor activity, and we really have run the study in triplicate in essence. Design of the phase 2b trial is a standard double-blind randomized placebo-controlled study, obviously in patients with moderate to severe pruritus. They qualified with moderate to severe during a 7-day run-in period where we had repeat measures of their worst daily itch scores. They had to have a baseline score above 4 on a 10-point NRS scale to qualify. They were randomized, as I said earlier. We had 3 doses of 845 administered intravenously 3 times per week, TIW, to these patients. Dialysis patients routinely receive 3 dialysis sessions a week. The 3 doses were 0.51, 1.5 micrograms per kilo versus placebo. 8-week treatment period, primary endpoint, mean change in their weekly worst itch scores from baseline at week 8, and then secondary endpoints related to quality of life measures. And we used these validated self-assessment scales that we're also using in our phase 3, Skindex-10 and 5DH. And we looked at other aspects of quality of life using standard measurements also. So overall, on slide 14, how does our drug perform versus placebo? So this is all drug treatment versus placebo, drug in purple, placebo in gray. We see that both groups start essentially at the severe level, a 7-point NRS score at baseline. By the end of the 8-week treatment period, the average 845 treated patient is into the mild category, whereas placebo has plateaued really after week 4 in this study. So a significant response for the average patient over the 8-week treatment period. When we look at the predefined primary endpoint on slide 15, left-hand panel here, mean change in worst itch at week 8, difference from placebo being approximately 70% improvement over placebo, obviously highly statistically significant. When we look at that particular change by responder rate, as illustrated in the right-hand panel for the lowest dose we used in this trial, the 0.5 microgram per kilo dose, at 8 weeks, 845 treated patients showing at least a 3-point response were 64% of the treatment group. versus 29% in placebo. So highly statistically significant difference. And this indeed is the endpoint we will be using in our ongoing phase 3 trials. Responder analysis at the 3-point level at this dose, 0.5 micrograms per kilo. Now there is no, as I mentioned, other labeled product or standard of care for these patients. What these patients tend to use and self-administer is generic agents such as generic antihistamines or in some cases corticosteroids, both of which we allowed in this phase 2 trial if the patient was on a steady standard regimen coming into the run-in period. And obviously we stratified those drug users across our various treatment groups. So what we're looking at in slide number 16 is we deconvolute the 845 response, looking at patients who had concomitant so-called generic antipyretics on board, and those without. Essentially we see no difference in the response rate there. So this is telling us two things. First of all, those agents are really ineffective in treating that pruritus. They're qualifying for the trial with moderate to severe, and the action of 845 is not influenced by any concomitant medications that these patients are taking. How effective is the change in pruritus over that 8-week treatment period as perceived by the patients? If we look on slide number 17, PGIS assessment, you see when we initiate this trial at baseline scores in the left-hand column here, over 50% of the patients had recorded severe or very severe pruritus scores, and that's diminished to approximately 12% by the end of the 8-week treatment period. So significant changes in category over this 8-week treatment period. We think that will improve even further when we move to the 12-week treatment period as we're using our ongoing phase 3 trials. When we look at quality of life scores across the two self-assessment scales, Skindex-10, 5-DH, on slide number 18, we see essentially all doses of drug are equi-effective here, and all are statistically better than placebo. So perceived changes in NRS are recorded by the patients as improvement in their quality of life across various aspects. And if we look at that correlation directly on slide number 19, we see there's a highly statistical correlation there for both self-assessment scales, Skindex-10 on the left and 5-DH on the right. So improvements in NRS scores are recorded as large improvements in quality of life scores for these patients. In terms of safety, tolerability, in slide number 20, these are treatment emergent adverse events of greater than 10%. Really nothing alarm within this group. We didn't see any particular safety findings raised by our Data Safety Monitoring Board. I'll talk about our ongoing safety trials in just a second. What we did see here was non-dose-dependent dizziness, non-dose-dependent GI side effects, and some somnolence being the most frequent side effects here. So nothing of great concern moving forward into phase 3. Some examples then of how patients can respond to KORSUVA or CR845 in slide number 21. The top two panels are individual patients, one on a 0.5 microgram dose on the left, one on a 1 microgram dose on the right. The little orange circles are their daily worst itch scores, and the green diamonds here are their overall Skindex-10 scores. And you can see in both these cases, these are patients who moved from a severe pruritus level at baseline of between 7 and 8 to a baseline of 0 to 1 at the end of the 8-week treatment period. So very potent, efficacious, robust response in these particular patients. And I should say in terms of the frequency of patients we see having such a response, 25% of the drug treatment patients in this particular trial had a reduction from baseline to at the end of week 8 to between 0 and 1 in their NRS scales. So very robust response in the CKD hemodialysis patient. So moving that from phase 2 to phase 3, obviously it's our aim to maintain as much of the design elements as possible into phase 3 from phase 2, and we did so. So as outlined in slide number 22, this is the design of both our CAM1 and CAM2 phase 3 trials. Major differences here. We extended the treatment period from 8 weeks to 12 weeks in our phase 3 trial. As opposed to looking at 3 doses of 845 versus placebo, we now have a 1-to-1 randomization at the 0.5 microgram per kilo dose. And other than that, entry criteria and measurement instruments are exactly the same as those used in our phase 2. NRS for recording our worst itch scores, and QOL measurements with these standard validated Skindex-10 and 5DH scales. Where are we on our phase 3 program? Illustrated in slide number 23. CAM1, fully enrolled, will read out this quarter. CAM2, ongoing globally in Europe, Asia Pacific region, some US sites. Will read out in the second half of phase 3. We also have an ongoing open label study that we initiated in Q2 of 2017, almost fully enrolled at the 240 subject level. And greater than 125 of these patients are through 6 months constant exposure to the drug, and approximately 40% of those are through the 1 year constant exposure levels. We have ongoing safety assessments by a data safety monitoring board for this trial, the last of which occurred last month for this trial and ongoing phase 3. And we've had no modifications in our safety monitoring based on that advice. So we don't see anything unusual in these ongoing long-term safety trials. So for this particular program, as I mentioned initially, Krasuva injection for hemodialysis patients, we announced in Q2 last year that we'd signed a license agreement with Vifor Fresenius, which is a joint venture between Vifor and, of course, Fresenius, being the largest dialysis provider across multiple regions. This was really designed to aid in our commercialization of Krasuva injection in Europe, Vifor being a leading nephrology company there. But also within this agreement, it allows us to exercise a co-promotion agreement built into this with Fresenius Vifor here in the U.S., specifically for Fresenius clinics. So about 38% of the clinics in the U.S. are run by Fresenius, and after launch, we think that's going to build momentum for this particular molecule if we have Fresenius co-promoting this within those particular clinics. Cara is booking all sales across the U.S. for Krasuva injection. So that's where we are with our lead program, our IV formulation. Moving on to our development of oral Krasuva. And our lead program here is in CKD-associated pruritus, but in this case in pre-dialysis patients. So these are non-hemodialysis patients. And this is also a very significant opportunity. Per NKF numbers, CKD is certainly under-recognized. And per their numbers, there are approximately 30 million CKD patients in the U.S. Per our analysis using IMS numbers, there is approximately 7 to 7.5 million CKD stage 3 to 5 patients in the U.S. And of that number, approximately 33% are using a prescription as an antipruritic agent, again, predominantly corticosteroids. So approximately 2 to 2.5 million pre-dialysis CKD patients experiencing pruritus to the level they're seeking antipruritic treatment. Again, no approved FDA therapies for these particular patients for this indication. In terms of reimbursement, unlike the IV formulation, the oral formulation doesn't fall within the ESRD bundle. So this, of course, is going to be predominantly commercial reimbursement for the oral medication. Where are we in the development of that? We announced last year that we'd completed a phase 1 study specifically within CKD stage 3 to 5 patients to look at the exposure levels we can attain with orally administered Krasuva. And illustrated in the bar chart here is the 3 tablet strengths we've taken forward into phase 2B, 0.25, 0.5, and 1 at the steady state overall AUC versus the AUC we observe with a 0.5, obviously highly efficacious dose we use with hemodialysis patients. And that's indicated by the blue dotted line here. So really, with all three of these tablet strengths, we should have more than enough exposure to see full activity at the human kappa receptor. Design of this phase 2b trial really mirrors the trial we're using in the phase 3 hemodialysis patients. Again, obviously double-blind randomized placebo control. We have a 7-day run-in to assess basement score. We randomize. We're looking at 3 doses of drug, 0.25 tablet up to 1 mg tablet, QD in this case. Again, example of the chemistry here, because this drug is eliminated via the kidney. We only need to see administration once a day for appropriate exposure. The 12-week treatment period, primary endpoint, change from baseline to week 12 in their NRS worst itch scores, and then the same instruments to look at QOL measurements, Skindex-10 and 5-DH. Other indications and clinical populations we're interested in for our oral formulation. 2 of those in particular, we've already run a phase 1 trial in chronic liver disease patients of all categories, essentially. And a phase 2 trial will be initiated this year in chronic liver disease associated, this quarter in chronic liver disease associated pruritus. And then finally, we announced last month that we'll be initiating a phase 2 trial in atopic dermatitis, and that will come around mid-year, with both these trials reporting out in 2020. Getting back to the advantageous characteristics of KORSUVA versus standard opioid drugs, as I mentioned initially, due to its chemical structure, we don't see CNS exposure. We certainly don't have a pharmacology of a classical opioid drug. Per FDA guidance, we have run a human abuse liability trial at dosages up to 30-fold higher than the dose we're using in our ongoing phase 3 trials with KORSUVA injection. What you're looking at in slide 31 is the overall drug liking scores from these non-addicted polydrug abusers that we use for this type of trial versus a standard Schedule 4 compound as recommended by CSS at the FDA for this particular trial. And you can see both dosages of 845 bolused into these patients pretty much look like saline, no statistical difference. And our Schedule 4 compound is recording a score of 73.3 on the liking scale. So based on this data, we would propose that 845 or KORSUVA injection should have a Schedule 5 or non-scheduling designation. So let me finish on slide 32 with the upcoming milestones through the rest of this year. Q2 sees our top-line data from our CAM1 pivotal trial. Second half of the year for KORSUVA injection, we have our second readout, our global phase 3 pivotal trial. And then for oral KORSUVA, we're going to see top-line readout from our phase 2 trial ongoing in CKD AP patients stage 3 to 5. And we'll be initiating 2 further phase 2 trials this quarter and around mid-year for chronic liver disease-associated pruritus and atopic dermatitis. Lots and lots of clinical activity and readouts in the next year or so. And I think we have the balance sheet to support that currently. We ended last year with $183 million in cash, approximately, and we've guided that takes us through into 2021, completing all of these clinical trials that I've just summarized. So thank you very much. I'll stop there. I see we have 5 minutes for Q&A, and I'm very happy to take any questions, so thank you. We're going to give top-line data as we normally do for major trials. Not the detailed analysis of everything within that. We want to publish these phase 3 data and obviously a recognized peer-reviewed journal at the end of the day. But we're going to release our primary and major secondary endpoints. Yeah. So the point that Alan's getting to, we have breakthrough designation in the past that has met with favorable responses from various divisions in the FDA, not so much the derm division, but some others, that with a breakthrough designation for an unmet need, it may be possible to file with one pivotal trial or reduce safety exposure numbers. That's a conversation we're going to have with the FDA post our first phase 3 data. So that's going to come in the second half of 2019. And if there is changes to the guidance based on that interaction, we'd certainly announce that. It's unlikely that we'd have a benefit from proven efficacy with one trial versus two, since we will have both readouts in a relatively short space of time. What would be a benefit is reduced safety exposures overall. Of course, current guidelines is 1,500 overall, with 300 of those at 6 months and 100 at 1 year. If we could see a reduction in the overall number, I think that would be beneficial for us in terms of timing. It's coming great. Everything's in line. Yeah, so we're at the very initial stages of thinking about commercialization here. We have looked at projected sales force sizes. That would probably be in the region of 60 to 75 reps for the U.S. market for KORSUVA injection. Preparation is underway. Obviously, the execution on a lot of that is going to be dependent on positive data as it comes along and as we look at the timing for the NDA filing. But yeah, we're well underway on the preparation for that. Again, I think helpful with the Vifor Fresenius deal that we're going to get some help in 38% of the clinics in the U.S. via that deal and the co-promotion within Fresenius clinics. So that helps us with the initial launch. But beyond that, we're going to market to the rest of the dialysis centers in the U.S. We do have an interim on and I neglected to indicate that. We do have an interim conditional power assessment on our second pivotal, and we will announce the results of that interim when we have it, as we did with CAM1. And I should also say we have an interim power assessment built into our ongoing phase 2b trial with oral KORSUVA for CKD stage 3 to 5 patients. And we will also announce the results of that particular interim assessment. So right now that trial is designed at 240 patients, 60 per group, and we have a possibility to double the sample size in that particular phase 2 trial, if necessary. Thank you very much. Thank you, Alan, for the invitation.